Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear students, in the previous lecture, we have already initiated one topic of this chapter, that is gene regulation, or we can say regulation of gene expression. And in that, we have already discussed about the points where regulation occur in case of the eukaryote and prokaryote, and also we have discussed about the lac operon. Now, today in this lecture, we are going to talk about the trip operon, tryptophan operon. Okay. So if we talk about tryptophan operon, this tryptophan operon is a repressible operon system. It is a repressible operon system. So if we talk about tryptophan operon, which is also known as tripoperon, here in this, it is known that three enzymes are necessary. Three enzymes are necessary for synthesis of tryptophan amino acid. And these enzymes are synthesized by activity of synthesized by activity of five different genes. Five different genes in a coordinated manner. In a coordinated manner. So this is the tryptophan operon. Now let's discuss first the operon with the diagrammatic pattern. See, if we talk about this operon, and if we draw the diagram of this tryptophan operon, it would look like this. Where this is the R gene, which is known as regulator gene. Then here you will find a operator gene, and then you will find a promoter gene. So here it is a promoter, it is operator. Now here trip E, trip D, trip C, trip A, trip B and trip A gene are there. And this all are the structural gene, that is from trip A to trip E. These are structural gene. Now if we talk about regulator gene form its mRNA, and its mRNA is going to form an inactive repressor that is known as aporepressor. So this is aporepressor, and aporepressor is inactive repressor. So inactive repressor cannot bind with the operator gene, and if it is not able to bind with the operator gene, then RNA polymerase will recognize the promoter sequence and synthesize all the further genes. So this trip E is going to form a polypeptide E. This trip D form a polypeptide D. Trip C form polypeptide C. Trip B form polypeptide B, and trip A form polypeptide A. Now we know that this E and D polypeptide combinedly form enzyme 1. This trip C is going to form enzyme 2, and B and A is going to form enzyme 3. Now all these three enzyme convert substrate into A, then enzyme 2 will convert A into B, and this enzyme 3 will convert B into tryptophan. Okay, so this tryptophan is going to synthesize. So when aporepressor is inactive and it is not binding with the operator gene, the structural gene are getting expressed and is going to involved in the synthesis of tryptophan. So this is in the presence, in the absence of repressor, active repressor, and it is a switch on condition. Now, how this becomes switch off? When tryptophan bind with the aporepressor, then it will be aporepressor become active. So when aporepressor become active, then it will inhibit the synthesis of this polypeptide. So now this can be shown like this in a diagram. Now here R is regulatory gene, P is the promoter, O is the operator, trip E, trip D, trip C, trip B, and trip A. Now here this is operator, this is promoter. Now here the repressor regulatory gene going to form a mRNA, and mRNA is going to form this aporepressor. Now when the tryptophan bind with this, which is known as co-repressor. So this aporepressor and co-repressor combinedly known as active repressor. So this active repressor is going to bind with the operator. And if it bind with the operator, the transcription of this particular structural genes get inhibited. And if this is inhibited, these are not going to form their polypeptide. And tryptophan synthesis will stop. So tryptophan itself acting as a co-repressor and making an inactive repressor, an active repressor, and so the synthesis of tryptophan is getting inhibited. So it is a switch off mechanism. So switch off is also made by the tryptophan itself. So it is a type of feedback inhibition. Okay, so it explains feedback repression. Okay, so this is the thing. So this way the tryptophan actually get regulated. So active repressor is due to the fusion of the aporepressor with the co-repressor, that is a tryptophan. And this active repressor bind with the operator gene and block the function of RNA polymerase. So the transcription would not occur and tryptophan operon will be switched off. So feedback repression is a function when there is a no need of the end product, or tryptophan is not needed in much more amount. Hence, there is a no requirement of continuation of this anabolic pathway and operon stops the process. So this is the one of the important thing. So it is a replaceable operon, okay, and that is all about the what tryptophan operon. Now, two genes are there. About that, we have to note down that may ask sometimes. One is constitutive gene. This is just note point of view. Constitutive gene, if we talk, they are also known as housekeeping genes. Okay. Now this constitutive gene or housekeeping gene so constitutive gene or housekeeping gene, if we talk, these are those genes which are constantly expressing themselves in cell, because their product are required for normal cellular activities. Means these are the housekeeping genes or we can say constitutive genes which are constantly expressing all the time, and there is no regulation of these genes, then they are known as constitutive genes or housekeeping genes. If we see the example, then genes of glycolysis enzyme are constantly expressing, genes of ATPase, I gene of lacoperon that get constantly expressed. But another type of gene are the non-constitutive gene. If we talk about the non-constitutive gene, this non-constitutive gene are also known as or luxury gene. These are not always expressing so these are not always expressing themselves in a cell. They are switched on or off. They are switched on or off according to the requirement of cellular activities. So if we take an example, then the gene for nitrate reductase in plants than lactose system in E. coli. So ZYA gene. Okay, so this all genes which are not always explained, they are regulated, they have a switch on mechanism, then they are known as non-constitutive genes. Okay, so this is about the tryptophan operon and the particular genes what we have we can study. Okay, I hope you understood this. Thank you.